Uh, a very good morning and a warm welcome to all of you who have joined us this morning for this very special program, Exploring Science with Shanti Bhatnagar Prize Winners 2020. I'm G. Mahesh, a scientist with the Science Communication and Dissemination Directorate at CSIR, and your host this morning. Last month, or to be precise, on 26th September 2020, the Shanti Bhatnagar Prize winners of this year were announced. The objective of this program is to get to meet the Shanti Bhatnagar Prize winners of the year, listen about their work, and interact a bit with them through this virtual means. The program is being streamed on CSIR's social media pages on YouTube, Twitter, and Facebook. And do click the share or retweet button right now so that we have more people joining us. And I'm sure this is going to be an exciting 90 or so minutes. So this morning, we have with us two Shanti Bhatnagar Prize winners of 2020, Dr. Bushra Ateeq from the Indian Institute of Technology, Kanpur, and Dr. Subhadeep Chatterjee from the Center for DNA, Fingerprinting and Diagnostics, Hyderabad. I shall have the pleasure of introducing both of them in greater detail, but that will be a bit later. For now, a very warm welcome to Dr. Bushra and Dr. Subhadeep, and hearty congratulations to them on winning the Shanti Bhatnagar Prize. It is quite possible that many of you watching this program also joined us in the last program held on the 10th of October, which was the first program of the series. In the last program, we had a number of school students joining us, and it is quite possible we have some students present with us today as well. All of you may know the Shanti Bhatnagar Prize is the highest prize for science and technology in India, and within the scientific community, it is often referred to as India's Nobel Prize in the field of science and technology. Well, that kind of highlights how prestigious the prize is. So that is a wee bit about the prize itself. This morning, we have with us not two, but actually three Shanti Bhatnagar Prize winners, including Dr. Shekhar P. Mande, who himself won this prestigious prize in the year 2005. I'm sure most of you watching this program would know Dr. Shekhar Mande already. He is the Director General CSIR and Secretary DSIR. Dr. Mande has a great career behind him. And prior to taking charge as DG CSIR and Secretary DSIR in 2018, he was the Director of the National Center for Cell Science, Pune. Other than the Shanti Bhatnagar Prize, he has several prestigious awards and honors to his name. He's an inspiration for many young scientists, and he's very passionate about taking science to the masses, especially through these regular webinars in these COVID times. Well, that's a very brief introduction of Dr. Mande. Thank you so very much, sir, for joining us this morning. I will now request you to give your opening remarks.
Thank you, Mahesh, very much for the kind words. And it's a great pleasure to have with us today Professor Bushra Ateeq of IIT Kanpur and Dr. Subhadeep Chatterjee of CDFD, Hyderabad. There is the Center for DNA Fingerprinting and Diagnostics, Hyderabad. I have personally had the pleasure of knowing both of them for last several years, and it is a great moment of pride for me that both of them have been awarded this year's prestigious Shanti Bhatnagar Prize. The Shanti Bhatnagar Prize is revered in Indian science and technology fraternity very much. It's considered to be the highest recognition in India, and society also looks forward to these scientists for their celebration, for celebrating their achievements, and for many more in the years to come. So I would just like to tell Bushra and Subhadeep that just the beginning, and what society would actually look forward from both of you in the coming years is many more fundamental contributions from your labs that are emerging. But at the same time, I am very proud that both of you have been recognized for the efforts that you have been doing in India in your own laboratories in the respective works. This is a series that we have started to let people know what the awardees actually do in their lab. And this year being a very special year, the entire series has been conducted online, as Mahesh said. But as also Mahesh said, please be aware that there might also be very young audience. So you may have to tone down your lecture so that everyone else can follow what you do. And as you can see that most people very eagerly look forward to hearing both of you and your contributions. So I will not stand between them and you anymore. And I'm myself looking forward to hearing both of you about your own work. Thank you very much, Mahesh, and back to you. Mahesh, you're mute.
Thank you very much, sir, for this nice opening remarks. Dr. Bushra's presentation is already up, as we can see. But before I introduce and invite Dr. Bushra, let me inform you that we have a question and answer session after both the talks. You may post your questions in the chat box as they occur to you. You don't really have to wait for the talks to end to post your question. And at the end of both the talks, I shall take up the questions. Also, please do share, retweet if you haven't already done so. When we discuss about diseases in the recent times, it is COVID-19 that immediately comes to our mind. Scientists and researchers all over the world are looking for a drug or vaccine for COVID-19. While COVID is a new disease, cancer has been around for centuries, and yet it is a disease which still does not have a so-called silver bullet. It is reported that there are over 100 types of cancer that affect humans. In the early times, there have been attempts to cure cancer by surgery, radiotherapy, or chemotherapy, or a combination of these. It is only in the 1970s that it came to be known that cancer is caused by the alteration in our genes. Since then, researchers and scientists across the world have been trying to understand the molecular basis of cancer. We have one such scientist this morning who is exploring the genetic and epigenetic changes that cause cancer and spread the cancer in an individual. A PhD from Aligarh Muslim University, Dr. Bushra is currently an associate professor at the Molecular Oncology Laboratory in the Department of Biological Sciences and Bioengineering at IIT Kanpur. She's also a senior fellow, DBT Wellcome Trust India Alliance. Earlier in her career, Dr. Bushra served brief stints as a research associate at AIIMS and the National Institute of Immunology, New Delhi. Then she moved to McGill University, Montreal for her postdoctoral training and later joined Michigan Center for Translational Pathology at the University of Michigan, where she also served as a research investigator. In February 2013, she joined IIT Kanpur, where she and her team are exploring the molecular events that take place in our cells that drive cancer. Last month, she was bestowed with the Shanti Swarup Bhatnagar Prize for Science and Technology in Medical Sciences discipline. Many congratulations once again, Dr. Bushra. Great to have you with us this morning. And so over to you.
So thanks, Mahesh, for such a generous introduction. And I would like to thank CSIR and the SSB jury for recognizing our efforts by bestowing such a prestigious Shanti Swarup Bhatnagar Prize to our group. And also, I'm very thankful to Professor Mande for such a nice introduction. And this is a great initiative by CSIR to start this series exploring science, which will connect scientists to the wider public. So, as Dr. Mahesh already kind of gave a brief introduction about cancer. So almost all of us have a relative or a friend or an acquaintance who succumbed to cancer. And of late, there's a steep surge in the cancer cases. And one good thing is most of the cancer, like around 90 to 90% of the cancers, they are lifestyle disorders or maybe due to environment. Most probably, when I say lifestyle, is obesity or the kind of food we eat or smoking or use of tobacco and so on and so forth. Only 5 to 10% of the cancers are related to germline disorder or hereditary in nature. That means they run in the family. So if you go back and look for the earliest evidence of human cancer, and we could trace back to ancient Egypt, where there was an incidence, this recent report, where they have shown one mummy, which is like 2,150-year-old, and the CT scan of that mummy showed metastasis in the lumbar spine region, which indicate oldest case of metastatic prostate cancer in ancient Egyptians. So it's very old. Though it's a lifestyle disorder, but the origin is traced back to so many thousands of years. And the first person to identify and name cancer was Hippocrates, and he named this disease as a karkinos, is a Greek word for cancer. And he also hypothesized that there are like 4 kind of fluids, like yellow bile, black bile, phlegm, and blood, and there is an imbalance between the 4 elements, and which give rise to cancer. And later on, there are like so many theories about cancer, so I'm not going to go into details about those theories, but I'll move forward. What makes normal cells different from the cancer cell, and what features cancer cells they have? So as we all know, the normal cells, they divide when it's required. So there is a check about their multiplication, and they only divide when it's needed. And they also stick together, means they are glued together, and they know where to stay. They stay in the right place. Sometimes when they undergo like some damage or they get old, they also destruct themselves, a term known as apoptosis. They become specialized or mature. So we know embryo, how it develop, and then different kind of cells are evolved, like bone cells or like hair or skin or different organs. So they mature, they specialize. Whereas cancer cells, they doesn't have any control. They just keep on growing and keep on multiplying. And they also ignore signals from the other cells. That's one feature. They don't glue together. They don't stick together. That is one very critical feature of these cells. They don't specialize, and also the damage they undergo, they won't repair, and they won't die that easily. So you can see here in the schema, this blue is showing some cancer cells, and you can make out like how different they are in morphology, and the nucleus is also slightly enlarged. Now, what makes cancer cells so different than the normal cell? So if this is the karyotype, like arrangement of the normal chromosome, the normal cells, this upper panel, and we know like in human cell, they're like 23 pair of chromosomes. So here you can see these 22 chromosomes, 22 pair, and this is the sex chromosome. This is from a male individual. That's why you see X and Y. And each chromosome is stained with a specific dye to highlight the orderly nature of these cells. So each chromosome is nicely distinct in different color. But when you look at the chromosomes from the tumor or cancer cells, you see a lot of like aberration. There are a lot of like chromosome instability. So when I say chromosome instability, means some chromosomes are divided or multiplied as a whole, and sometimes one branch or one arm of one chromosome is getting fused with other chromosome, giving rise to a chimera. So how this happens? So if you look here in this schema, I have shown like sometime what happened in a chromosome, a small chunk of chromosome get deleted, and then it gives rise to a shortened version of a chromosome. Sometime a small piece of chromosome get duplicated, giving rise to a longer chromosome. Sometimes just simply inversion, so it's upside down that region. And another occasion, there's an insertion, a piece of a chromosome get inserted in a different chromosome, giving rise to a chimera gene or like a chimeric protein. And in some cases, there's simple translocation. A piece of chromosome 1 or 4 like is getting rearranged with 20, and you can say there's a swap over of the chromosomal fragment. So these are different aberrations that take place in the cancer cell karyotypes. Now moving to like how these cancer chimeras, chromosome chimeras dictate the cancer cell. So this is a mass, tumorous mass from a patient. And you can see there are like 2 different kind of cells. So you can imagine like each cell in a cancer tumorous mass, they have different kind of chromosomal aberration or mutations. And that like aberration, they make this cancer cells very distinct. So here in this schema, I have shown like orange and the green cells. So this is the heat map about different drugs which could work on these cells. So you can make out like green cell here is only sensitive. This purple and blue is showing the sensitivity. So green cell is sensitive to drug 3 and 4, but it's resistant to drug 2. Likewise, this orange cell, this is sensitive to drug 1 and 2, but resistant to drug 4. So that's why this is the reason when the patient, cancer patient, they get a therapy. Some patient may respond temporarily and then again the cancer relapse. Some patient they don't respond to cancer drug at all. So this is the reason there's a disparity. There's like a lot of like distinct molecular aberration in each cancer cell, and that term is also known as heterogeneity. And because of this heterogeneity, the treatment is the biggest challenge in these days. So now Moving to the prostate cancer, because that's a title of the talk. So prostate is a small gland which is almost the size of like walnut, and it's located at the base of urinary bladder. You can see here, and this is the normal section of the prostate gland, and here you can say cancerous gland where there's a small tumor in here. The normal function of the gland is to secrete the seminal fluid and also nourish and protect the sperm. Although the growth and the maturation of the gland happen only when the males they hit the puberty, and that is because the maturation is totally dependent on the androgen, that is like male hormones or testosterone. So one indicator or biomarker that is usually used in the clinic to detect prostate cancer is PSA, that is prostate specific antigen, and the normal level is 4 nanogram per ml, or lower than that it's considered normal. And but again, this PSA level is not a gold standard test, because many times this give rise to false positive and false negative results. Now, what happened? Like, this is the gland, and you can see this is one foci from the gland, which is shown here, and there are thousands such foci. So this stage is benign; it's not cancerous. You can see all cells are nicely arranged. But once there is an oncogenic hit, there's some aberration in these cells start dividing, which is shown here by blue and yellow. And this stage is known as high-grade prostatic intraepithelial neoplasia. But once these cells keep on dividing, as I said, because they lose the control, and the cells leave this foci and they go into the other foci, but within the gland. So they are localized in the prostate gland. So this is known as localized prostate cancer. Now, what happened over the period of time? They change the phenotype, they undergo EMT. So EMT is mainly like epithelial to mesenchymal transition, where so in epithelial cells, they are more glued together, they are more like, they are not that motile, and mesenchymal feature is more motile. So what happened, these cells which acquire the mesenchymal feature, they metastasize. So when I say metastasize, they move from the origin of organ, that is prostate, to other organs that such as bone, lung, or liver. So that is known as metastasis. As I told you, there are like numerous genetic aberration, and in this chart, you don't have to like worry about what is written here, but you can make out there are like so many different genetic rearrangement of chimeras, one gene getting fused with the other gene. So there are like more than 25 different kind of genetic rearrangement in prostate cancer. But in today's talk, I'm going to only focus on two. One is like TMPRSS2R. And what happened here, the small chunk like exon 1 here, a small chunk of TMPRSS2 gene is getting fused with this gene, ERG, and it gives rise to this kind of chimera fusion. This fusion is recurrent in 50 to 60% of the prostate cancer patient. And because of this fusion, it gives rise to ERG oncoprotein, which is otherwise in a normal prostate is not present. The other subtype, which is also highly expressed in prostate cancer patient, is SPINK1. It's a serious protein inhibitor, casal type 1. Again, these patients who have SPINK1 higher expression, they show very aggressive disease, and they are like as advanced stage patient, and they also exhibit like poor clinical outcome. Now, moving to like when I started in 2013, as Mahesh mentioned, so one of the biggest challenges was what are the recurrence of these major subtypes in Indian prostate cancer patient, and this data was not known. So this was a big thing that the foundation was missing. But you can make out here like in Caucasian is 50% TMPRSS2, in Chinese is half. So there is some racial disparity between these genetic rearrangement. Also, like you can make out like here this PTEN deletion, which is a tumor suppressor gene, was also less in Chinese and Japanese. So to fill this gap, what we did, I started collaboration with several medical colleges. So with local medical college in Kanpur, GSVM, and King George Medical University, KGMU Lucknow, AIMS New Delhi, and a private pathology lab in Gomtinagar, Lucknow. So what we did, we collected about 121 prostate cancer samples, which were procured by doing different kind of surgeries like prostatectomy, where you remove the entire gland, or needle core biopsies, where you use needle to take out biopsy, or TURP procedure. And all these specimens were subjected to very traditional immunostaining or RNA in situ hybridization, where you use RNA probe to detect the presence of that RNA, or by fluorescent in situ hybridization. So I'll tell you what exactly these are. So again, this is a patient where like we use prostatectomy specimen, and this brown stain is indicating the presence of ERG oncoprotein. Then we kind of figure it out like whether this is because of genetic rearrangement or not. So you can see here, this is the ERG gene, which is located on chromosome 21, and there are two different probes, red and green. So you can see in the microscope, if the probe are together, that means they are giving a signal green and red together. That means the locus, that gene is intact, there is no breakage. But once these probes are separated, they are separated out. You can see green and red separated out. So that means this there is a rearrangement about this locus. So what we found, it was a first report from a northern part of the India, that 49% of the prostate cancer patient, they harbor this genetic rearrangement, and almost all the benign cases which were non-cancerous, they were negative for this genetic rearrangement. Now, next thing is like we screen another patient where we collected a sample through needle core biopsies. So you can see there are 12 different regions from there the biopsy is taking, because it's a round gland and they want to figure it out which part of the gland has the cancer. So there are like 12 different cores. And when we stain these cores, we can see this brown staining, and brown staining here denotes the presence of a SPINK oncoprotein. And this is the needle core biopsy. This is how it looked like in the microscope. And again, we found around 13% of the prostate cancer patient, they were positive for SPINK1. One important thing was all the SPINK1 positive cases were negative for T impresses 2 rearrangement that I've shown in the previous slide, therefore showing some kind of mutual exclusivity in terms of genetic alterations. Now, after doing all the analysis, we kind of filled this table. So these are the different aberrations that we found from 121 patients from northern part of the India. And what was striking was this RAF rearrangement was about like 5.5%, which is good because there are many FDA-approved drugs which are used in the melanoma for BRAF mutations, such as sorafenib, could be used for this patient. The other thing was like, majority of these SPINK1 positive patients, they show aggressive disease and poor prognosis. So you must be thinking why we need to go at this level, why we need to figure out what are the genetic alteration in the prostate cancer patient. Yes, the reason is clear, because once you know what are the oncogenic alteration, what are the pathways which are activated, you could decide the treatment based on that. As I mentioned about RAF rearrangement, you also know the fate of disease. If they are SPINK1 positive, that means they are progressing faster, they're aggressive kind of cancers. You also know what kind of therapies will work. So that's why it's very important to molecularly categorize patient based on the genetic alteration or the pathways which are activated. Now from here, I'll move on to the therapies. So one of the mainstay therapy in prostate cancer is androgen deprivation therapy, also known as ADT. So in 1940, Huggins reported for the first time when he castrated like older dogs, like surgically castrated older dogs, he's observed a rapid shrinkage of the enlarged prostate gland, and that kind of gave him an idea like maybe they're dependent on androgen signaling, on testosterone. So Huggins and the Huggins, like they showed that surgical castration in men with prostate cancer really benefited them. So for their discovery in 1966, Huggins received Nobel Prize in Physiology and Medicine. And since then, like this is the therapy which is mainstay and used in the clinic, irrespective of the molecular subtype that I showed you before. But because of the like emotional, psychological issues and the social stigma, there was a surge in the there was a surge in the chemical castration, because with the advent of this therapy, like people started looking for a small molecular inhibitor which could be used instead of surgical castration. And these are the ways how people do chemical castration. So either by using LHRH agonist, which is luteinizing hormone, releasing hormone, or by using anti-androgen therapy. As I said, that means you are blocking the action of androgen or male hormones in the body. So there are two ways. So when you use LHRH agonist, you kind of like inhibit the production of testosterone in the testicles. The other way of using anti-androgen. So when you use these molecules known as anti-androgen, you kind of inhibit the action of testosterone on the prostate gland. So these are the kind of ADTs, like chemical castration, which are used currently in the clinics. So how does it work? So as I told you, androgen receptor could interact with co-activator, which will activate its activity, or with co-repressor, which will inhibit its activity, and then modulate the expression of target gene. So here in this schema, you can see there's a testosterone. It will enter the prostate cell, get converted into more potent form known as dihydrotestosterone, and then it will get bind to AR, which is like have a pocket for DHT. And as a result of that, two AR molecule or proteins will bind together, form a dimer, and then get entered into the nucleus. And then the nucleus, they go and sit on the target genes. And since they are like driving these target genes, you can see PSA that I mentioned in my previous slide, as well as TMPRSS 2. There will be increase in the expression of these two, which will further support the growth and the survival of prostate cancer. And that's the reason most of the time, like the ADT, the chemical castration is used against either this testosterone or at the testosterone level where it's being produced. This is the chart to show you how it works. So on the y-axis, you can see there's a tumor volume and activity, and the x-axis, you can see the timeline. So here the patient is diagnosed with prostate cancer, underwent surgery and the radiation. And at this stage, the cancer is hormone sensitive. So when I say hormone sensitive, means patient respond to androgen deprivation therapy, that ADT I mentioned. And as a result of that, you see drop in the tumor volume as well as activity. Once you give the therapy, after some time, they enter into pre-metastatic stage, and that is due to selection pressure of the ADT drugs. And once they enter this stage, they keep on growing. They are no longer responding to ADT therapy, and they are known as castration-resistant or castrate-resistant prostate cancer. So at this stage, they're really aggressive. They won't respond to this ADT therapy, and the metastasis is also radiographically very evident. So you can make out by X-rays that cancer has metastasized to the bones. So at the cellular level, what is happening here? This is the foci that I showed you in the beginning. So these are the cells, and you see green cells are oncogenic cells with a more oncogenic property. They keep on dividing, and this stage is known as adenocarcinoma. Once you give androgen deprivation therapy, at some time, like they respond, they show decrease in the cell division, but then they overcome this and they start behaving differently. They turn into castration-resistant prostate cancer cells, and this cell, which is differently looking, this is like more of a spindle shape, is known as neuroendocrine prostate cancer, which is due to failure of this treatment. And you see like multiple of this spindle-shaped cell, they divide, forming neuroendocrine prostate cancer. And in this stage, the cancer is totally untreatable. They're like most of the drugs, they won't work, though people give chemotherapy and kind of combination therapies at this stage. So this is highly aggressive stage. So what we did in our lab to figure out the connection between SPINK1, which is highly aggressive subtype, and androgen signaling, we stained a tissue microarray, and this is a picture of tissue microarray. Each circle here is showing a tumor biopsy from one patient, and we had like around 237 patient samples. And then we stained these samples for two protein, SPINK1 and AR, that is androgen receptor. So this is the biopsy from patient 1. You can see it's negative for SPINK1. There's no brown staining here, whereas here you can see there's a brown staining. There's a positive for AR expression in this case. On this panel, you can see this is patient 2 is positive for SPINK1 expression, whereas the AR expression is very mild, it's moderate. So when we put data together and we kind of analyze, we found most of these SPINK1 positive patients tend to have either low or negative expression for AR, whereas SPINK1 negative patients, they tend to have higher or medium level of AR expression. So it's kind of showing inverse relationship between SPINK1 and androgen signaling or androgen receptor. So now I'll again show you what exactly happens. So here you have androgen receptor, and in order to get activated, it requires testosterone, that is dihydrotestosterone here, or synthetic androgen. And but if you give ADT, then it won't be getting active. So what we did in the lab, we used two different kind of cells, prostate cancer cell, one with higher SPINK level, the other line with lower SPINK level. So you can see in this schema, in this picture, we treated these cells with synthetic androgen, and as a result of that, this is the different time points, and you can see in 2 days at 36 hours or 48 hours, there's a decrease in the SPINK1 level, and which is also evident here by the loss of this green signal at 36 and 48 hours. And in this experiment, what we did, we used VCAP cells, which is like harboring like lower expression of SPINK1, and we treated these cells with enzalutamide, which is an ADT drug used in the clinic for prostate cancer patient. And you can make out how when we treated with this drug, even as low as 1 micromolar will trigger the expression of SPINK1 oncoprotein, which is evident here as well. So you can see more SPINK1 in the cells which were treated with enzalutamide. So it's kind of showed like there's an inverse relationship, and you could modulate this androgen signaling to alter the SPINK1 expression. Now, by doing several experiments in the lab, like ChIP-PCR and ChIP-seq, what we showed that AR and the rest, they both team up together to silence down or turn off SPINK1. So you can see, but when there is a loss of rest, the partner of AR, androgen receptor, that initiates the start of NEPC. Now, what SPINK1 does, once the AR and rest they are missing, rest is missing, SPINK1 partner up with the SOX2, and then SOX2 acts as a transcriptional activator, and then initiate the expression of SPINK1 and reprogram cells and drive towards NEPC. So this is what we found by doing several experiments, and this is how it looked like when we look at the in the context of DNA. So AR and rest, they get recruited on the AR target genes, that is SPINK1, and as a result of that, there's silence, and as a result of that, you see there's a decrease in EMT, stemness, and chemoresistance. But what happened when you use ADT, when you use anti-androgen, like I showed you, enzalutamide, it won't get recruited on ARE and RE elements, and as a result of that, SPINK is on, and because SOX will get recruited here and transactivate its activity, and you see there's an increase in EMT, stemness, and chemoresistance. Now to figure out what does SPINK do, so we performed like an assay which will tell us whether cells are harboring this stemness or not. So this panel here is showing normal 22RE1 cells which has higher level of SPINK1, and these two cells are genetically modified to silence SPINK1. So this is an assay to assess the stemness, known as like a prostate sphere assay, or you kind of measure the spheres. So these spheres are having like millions and millions of these cells. So the larger the sphere, the more the stemness the cells are having. And when we silence SPINK1, you can see the mean area of the sphere was reduced, or also the efficiency of forming sphere formation was also hampered. Now, in another set of experiment, we look for another marker, that is like stemness marker, LDH, aldehyde dehydrogenase, and we looked for its activity, and we found when we silence SPINK1, in this case, you can see here, the LDH activity was almost 50% reduced. That means that SPINK has some role to play in its stemness. So when you remove SPINK, you see there's a decrease in the stemness. And to figure out the connection with SPINK1 with EMT, we again prepared the protein lysate, and we run this gel to see the protein. So we can see when we reduce SPINK1, this is a SPINK1, when you reduce silence SPINK1, you see ECAT coming back, ECATrin coming back. And as I told you in the beginning, that this is very critical protein which glue cells together so that they won't move. And this is more present in the normal cells than in cancer cells. So you can see it's coming back, whereas vimentin, which is a marker for more aggressive cells or mesenchymal cell, was almost lost. Now we have shown that SPINK, which is like a hyperactive kid in a prostate cancer playground, will engage with stemness. It also moves cells from epithelial towards mesenchymal phenotype, which is much more aggressive and motile. It also keep on moving this cell cycle, make more cells, put them like a trigger to make cells proliferate faster. So this is indicate that SPINK1 has multiple role to advance prostate cancer. Now, to recapitulate the scenario that I mentioned about prostate cancer patient undergoing ADT or androgen deprivation, what we did, we took a line known as like Allen cap, and this line is negative for SPINK1, and it's also responsive. It responds to androgen signaling. So what we did, we grew this line in a condition that is known as androgen deprived condition up to 30 days. So you can see morphologically when the cells were underwent this androgen deprivation, there's a change. You can see this cellular projection coming out, also known as neuronal outgrowth, and see like it's keep on growing. Like at 30 days, they're like really large. So the cells were like isolated, and the protein was prepared from these different time points, and the page gel was run, and you can see here the SPINK1 was coming back. So at day 0, there was no SPINK1 because this line is negative, and the cells underwent androgen deprivation therapy, androgen deprivation treatment. We call them Allen cap AI. And the other thing is why it's coming back. So as I told you in the beginning, that SOX2 is a transactivator for SPINK1. It's activate the transcriptional activity of SPINK1. This switch is on, whereas REST, which is acting as a brake, was off. So REST is lost when this transdifferentiation. To figure out what does SPINK do in neuroendocrine prostate cancer, so what we did, we took again this Allen cap cell and genetically modified these cells by silencing SPINK1, and we took the cells which where you have higher expression of SPINK1, or the Allen cap cell where you silence SPINK1, and then these cells after ADT, like for 30 days, you can see they're having higher level of SPINK1, because as I told, the rest is lost and SOX is coming up to increase the SPINK1 level. And you see there's an increase in EMT as well as neuroendocrine marker known as NE-markers. And here, because SPINK is lost, because we genetically modified it, and you see decrease in EMT as well as NE-markers. This is the pictorial representation of these cells. And you can see here, when we silence SPINK1 in these cells, these AI cells, you can see the length of the cellular projection is reduced as compared to the control. So which indicate that SPINK1 play a critical role in the maintenance of this neuroendocrine phenotype in prostate cancer. So its role is very important for maintaining this phenotype. So now next, we kind of showed that in order to initiate NEPC, rest is lost. So we kind of figure it out that casein kinase is an enzyme which is responsible for degradation of rest. So then we thought like it's important to maintain the level of rest. How we could increase the level of rest at that stage so that SPINK won't come back. So we use casein kinase inhibitor, CK inhibitor, which will inactivate its activity, and as a result of that, rest is not degraded. The level of the rest will be maintained. So in this case, what we did, we use 2,2-RE1 cells, which are, as I told you, SPINK1 positive, and treated these cells with casein kinase inhibitor at different concentration. And as a result of that, as I told you, it will maintain the rest level. So you can see the rest level is again restored, it's coming back. And as a result of that, because rest is acting as a brake, so you can see decrease in the SPINK1. Then we also perform different oncogenic assays to assess the activity of casein kinase in these cells. And this is the graph showing cell proliferation assay. Blue is showing control cells and all these different concentration of casein kinase. And you can make out when the cells are treated with casein kinase inhibitor, the cells are dividing slower. They're not dividing as fast as the control cells. In another set of experiment, we perform foci assay. So when we treated these cells with casein kinase inhibitor, the cells were not forming foci, whereas control cells form multiple foci. Likewise, in the sphere formation assays, casein kinase treated cells didn't form like bigger spheres, and the efficiency was also hampered, whereas control cells where you have higher level of SPINK1, they form bigger spheres. So again, it indicates that you could kind of you could regulate the rest level, you could restore the rest level by using casein kinase inhibitor, which as a result will put a brake on the SPINK1 expression and SPINK mediated oncogenicity. So in order to recapitulate the entire thing in the mice model, because it's very important to test whatever we are showing is happening in the mice model, what we did, we designed an experiment. We used this immunodeficient mice where we implanted VCAP cells in the prostate gland of these mice and then let the tumor grow for 4 to 5 weeks. And at this stage, the PSA level in these mice was more than 4 microgram liter, that is like around 5 nanogram per ml. And at this stage, these mice were surgically castrated, and then they put into different groups where like vehicle control didn't receive any drug treatment, enzalutamide, and this is new generation anti-androgen ARN, also known as apalutamide. And these mice were treated orally for 4 weeks. And at the end of the 4 weeks, the tumors were excised out and stained, the presence of these markers that I told you. And what we found that the mice treated with enzalutamide and AR, they showed higher expression of SPINK1, which is evident here with the brown staining. Also, these markers like CYP, CGA, and TUB, these are the marker for neuroendocrine prostate cancer, were also coming up. So this experiment indicates, so when you put these mice under ADT kind of condition, you put tumor which were negative for SPINK1 or these neuronal marker, but over a period of time, these the tumor, they transdifferentiate into a neuroendocrine prostate cancer, which are much more aggressive. Similar data was also generated for the NEPC patient, which were underwent ADT, and there also we found higher level of SPINK1 accompanied with lower level of AR expression. So what we have shown in a nutshell that when you use ADT, that is androgen deprivation therapy, you tend to increase SPINK1, and SPINK1, since it's a autocrine paracrine growth factor, is associated with multiple oncogenic properties such as stemness, drug resistance, and cellular plasticity. And for cancer progression, cellular plasticity is very important because it initiates the conversion of cells from one lineage like adenocarcinoma to neuroendocrine prostate cancer. The take-home message from this brief talk is, if we administer a prostate cancer patient with ADT or AR-targeted therapy, that may result into increase in SPINK oncoprotein, which is accompanied with NE-marker, which leads to lethal kind of neuroendocrine prostate cancer. Therefore, it's very crucial to weigh the benefit of ADT against the possible side effect. And we, for the first time, also explored the paradoxical clinical outcome which is associated with ADT, and that is possibility to increase its SPINK1 level and its tumor progressive properties. We also proposed that casein kinase 1 inhibition could be used as a strategy to consider adjuvant therapies for these advanced stage patient or neuroendocrine patients. So there is a need to conduct a well-planned clinical trial using these casein kinase inhibitors. So finally, I would like to acknowledge my group members. So these are the bright and the wonderful people with whom I work. And the data I presented today was generated by Ritika Tiwari and Nishad Manzar. So Ritika already graduated. Currently, she's a postdoc at Cleveland Clinic, Ohio. And I'm very thankful to my all the collaborators, especially clinical collaborator, without whose help this conducting this work was totally impossible, and other collaborators abroad. And the work I presented today was funded by India Alliance Wellcome Trust, and I'm also thankful to DBT and SERV for their financial support. Thank you.
Dr. Bushra, for very lucidly presenting this complex topic of the prostate cancer, the work that you are doing, very extremely important work studying the genetic alterations in the Indian population. I'm sure there would be a few questions which we shall take up after Dr. Subhadeep's talk. For now, thank you so very much. You could stop presenting the slide so that
Sure.
Yeah, thank you so very much. So the next speaker, if you remember, those of you who had connected with us on 10th of October would remember that the program had ended prematurely, and in fact, just a little bit after Dr. Subhadeep had begun his talk, on that day the lease line had become unstable and had gone dead. But today, fortunately, it's holding well, at least so far. So, Dr. Subhadeep, thank you so very much for joining us this second time. It is my pleasure to introduce Dr. Subhadeep Chatterjee, a scientist at the CDFD Hyderabad. He obtained his PhD from CSIR PCMB Hyderabad in 2004 and he served as assistant professor at Shanti before proceeding for postdoctoral training at the University of California in Berkeley. He joined CDFD in 2008 as a staff scientist, where he and his team are working on interactions between plants and microbes. Dr. Subhadeep Chatterjee is a recipient of several awards and honors, including Department of Biotechnology's National Bioscience Award for Career Development, and most recently, the Shanti Swarup Bhatnagar for 2020 in the field of biological sciences. Congratulations to you, Dr. Subhadeep. So over to you.
Yeah, thanks. First of all, I would like to thank Dr. Shekhar Mande, DG CSIF, for giving me the opportunity to present the work, and for Mahesh for his coordination in this webinar. So what I will be doing in couple of minutes, 20-25 minutes or so, is to give you a glimpse of social language of bacteria, and I will take you to their world, like how they speak with each other, and whether microbes have a social life. And then I will talk about our work very briefly. Since I have been told that this will be a general talk, so I will not show you any data. So this will be just based on our findings and some of the earlier findings. So what the social life of bacteria looks like. So as you can see, in various life forms, response by communication is very key, one of the key features of all living organisms. And this is by doing so, they respond to change in the environmental condition. So that by communication, they respond to change in their environment and tell their fellow mates what to do next. So different forms of communication have evolved, and from blinking fireflies to song by whales, and there are dancing language of the honeybees. So different forms of life communicate with each other and tell their fellow mates about the changes in the environmental condition or whether there is some danger which is coming. So this communication has evolved to the highest level in the form of language, and many evolutionary biologists now think language as a living creature and compare it with the biological evolution. Like language, like biology, life is evolving, like language are also evolving based on selection and genetic drift. So since I told you that communication is very important, so how small microbes communicate with each other? If you see the life of a microbe, it's a very solitary looking organism, planktonic, no company with each other. That was being thought in early up to late 80s and 90s. But the question came, do unicellular solitary living bacteria communicate? They are very small in nature, and do they have a social structure or not? So the idea came by a very indirect serendipitous manner by some marine biologist. So there was some marine biologist working on a squid called as Hawaiian squid, which is found in the Pacific Ocean. So the squid has a unique property. So if you can see the arrow marks, so the squid emits light in the night, and this light actually helps the squid to find its food. So marine biologists got very interested and they wanted to find out where the light is coming out. And what they figured out, very surprisingly, that the light which the squid emits is not its own light. The light is emitted by a group of bacteria which colonizes the light organs of the squid. So naturally, the microbiologists got very excited and they started culturing this bacteria in the lab. And when they this bacteria in the lab, they found a very interesting observation that they found out that the light produced by the bacteria is only at very high cell density. So when they are very high in number, they will produce light. When they are in low in number, they don't produce any light. So that gave them a clue that there is something, some message or chemical which is secreted by the high density bacteria or large number of bacteria, which is being left in the medium, which helps them to communicate. So they did a very clever experiment in which they took the spent media from a talking bacteria and put it in a non-talking bacteria. And they found amazingly, the non-talking bacteria started to communicate. So that gave them the clue that there are some chemical communication substance or message which is secreted by the bacteria and which helps them to communicate with each other. Then the whole field of social microbiology evolved, and now this is a big field where bacteria undergoes transition of solitary to a community life form, and this is a reversible process. And while doing from a solitary to a community life, they form the structures which you see, a mountain-like structures. So these are biofilms, which you can observe in our lab. In our lab also we study biofilms. And these mountains or the buildings are nothing but built by the bacteria, by the secreted products of the bacteria. So they form a large gated community which has very nice drainage system for the inflow of nutrients, and also it helps them to protect against the hostile environment. So now I will give you the idea about the quorum sensing, what I talked about in the cell-cell signaling or chemical molecules. So the bacteria, as I told, secretes small signaling molecule, which are called a diffusible signal molecule, in a density-dependent fashion. So at low cell density, the amount of signaling molecule is less. At very high cell density, the amount of signaling molecule is also high. And when it reaches a particular threshold, it tells the bacteria what to do. So it tells the bacteria, now you are in large number, now you perform this task or social behavior. So indirectly, actually, if you can see, the bacteria can count each other and to determine what action they must take. And this action is taken by the expression of gene expression in an orchestral manner. So this is called as quorum. Quorum means a minimum number of members who are required at a meeting in a layman's language to have a decision. So as I told you that bacteria love to talk with each other, but you can't listen to their language, but you can see their language. So what we do, we develop biosensors which can detect the signaling molecules. Here is a GFP-based biosensor. And in the plate, you see lots of bacteria, they are talking with each other. So they are green. And in another plate, you see lots of bacteria, but they are not talking. So bacteria in which genetically modified not to able to speak. So when the social microbiology field evolved and further lot of research happened in the terms of bacterial signaling molecule, people found that bacteria makes very diverse form of signaling molecules. And one among the most common signaling molecule is by the acyl homoserine lactone. So it is very similar. I will give you an analogy to Indian languages. So like Indian languages, like in most of the Indian language has Sanskrit as a base. So similarly, in acyl homoserine lactone, lactone is the base, and then acyl chains are added by different bacteria to have different dialects. And we work on a family of signaling molecule, which I will come later, is called as diffusible signaling factor, which is a fatty acid-like signaling factor. And it is very similar to human communication. Like many bacteria are there, they can speak in multiple languages. And there are certain bacteria which can't speak, but they can listen to other languages. This is, for example, Vibrio cholera, which causes cholera disease, can speak in 2 different languages. And they can understand these 2 different languages. And as I told you, many bacteria, they don't speak or they don't communicate with language, but they have receptors on their cell wall or cell membrane, which can perceive the signal by the bacteria, other bacteria which are in around its community. It is very similar to our noisy neighborhood cartoon, in which neighbors are more interested on who have come rather than the person himself. So actually these bacteria keeps their receptors or ears open to know that who are our neighbors and how, based on our neighbors, we can change our behavior. So I will give you a glimpse of a very simplistic model of quorum sensing signal transduction. So in a take-home message, there is a synthesis enzyme which makes the signaling molecule in a density-dependent fashion. And then there are receptors which binds to the signaling molecule at a particular threshold level. And once bound, they bind to the DNA and regulate gene expression in a unison. So now people may ask, what is the advantage of talking with each other? So the advantage of performing social tasks is to achieve maximum benefit as a group. So I will give you a very simple example. Suppose there is one person who is firing bullets and there is a barrier. So the firing power is so low that the barrier may hardly break. But when multiple persons are firing the bullet at the same time, there is a great chance that the barrier may break. So this is what is called as the quorum sensing benefit, that there are certain tasks which are done in unison, which are always beneficial rather than the same task which is done alone. So in our lab, we work on a very important group of pathogens called as Xanthomonas group of phytopathogens. Xanthomonas is one of the most widely diverse plant pathogens, which causes very different type of diverse diseases in economically important crops, such as rice, vegetables, fruits, and many other economically important crops. We mainly focus in our lab on the rice and the vegetable pathogen, which is Xanthomonas oryza and campestris. So while working on this bacteria, we found out a quorum sensing signaling system, which we called as DSF, diffusible signal factor, like signaling molecule, which is secreted by the bacteria in a density dependent fashion. And the most important thing is that if you don't have this signaling molecule, the bacteria cannot cause disease. So this is the disease symptom which is caused by a wild type bacteria. If you knock out the signaling system or the language system, it loses the ability to cause disease. So we started figuring out in our lab how this signaling system works. What is the mechanism of this signaling system? So as I told you, you can tag this bacteria with the language molecule reporter system, and you can track this bacteria inside the plant. So this is the section in the plant in which the bacteria is colonizing in the xylem vessel. Xylem is like similar to human arteries and vein, where the nutrients and water can flow in plants. So coming in a nutshell, the take-home message is that after we perform a lot of molecular analysis of this bacteria, to understand its social behavior, how quorum sensing is influencing the social behavior. We found out that it has a very complex quorum sensing signaling cascade in which it perceives the signal which it makes in a density dependent fashion and then transduce the signal with collaboration with other two component system in the bacteria and to orchestra the expression of genes so that it gives a group benefit to the bacteria. So in a take-home message, it does a two works at a time. So at low cell density, when the quorum sensing signaling molecule is low, the low signal gives a type of signal to the bacteria to move and attack inside the host. So all the motility and chemotaxis is activated. Once inside the host, it starts synthesis of its virulence factor and they cause hydrolysis of the plant cell wall to release nutrients and the bacterial population grows in number. Once it grows in number, it tells the bacteria you form a biofilm. And how it does it so? Because it tells the bacteria to make lots of these attachment proteins to hold each other and with the plant cell. And this happens in a very orchestral fashion. So at low cell density, it tells the bacteria to move and attack, and at high cell density, it tells the bacteria to form a biofilm and to protect itself from the hostile plant environment. This also gives another advantage to the bacteria that if at low cell density, if it shows off all its weapons, then the plant may mount a defense response and kill all of them. But at high cell density, when it shows off its weapons, which are necessary for its virulence, then even if the plant mounts a defense response, 50% may got killed, but 50% will survive. So this is the idea about regulating its action based on its number. I will give you an example like how you can modulate the behavior of the organism by changing the signaling molecule. So this is a wild type cells. These are the bacteria which are moving. And now you see if you remove the quorum sensing system, so it moves very fast. So that means quorum sensing system, actually low quorum sensing system actually tells the bacteria to move towards the direction of the host. And when you spike in the quorum sensing molecule, synthetic molecule, then you see you can even stop the movement of the pathogen. So now this story has three parts. One is I told you about the benefit which it causes when you are in high in number and to perform a task together. But it is also having a double-edged sword. Imagine a situation in which this bacteria in a group, they are performing, and then there is the occurrence of cheaters. Cheaters are those bacteria which do not talk and they take benefit of the performers. So under this scenario, what will happen? Those who have prepared for the exam are working hard and the cheaters will take the benefit. And if this continues, the cheaters will be more in number because they outperform the performers, because they are taking the benefit from the performers. They are not spending their energy and the whole system will be with cheaters and the examination will collapse. This is the one problem of the talking together. The other problem with the talking, as I tell you, I gave you a hint that if you talk too much, you can alert the host. Then the host can mount a defense response on you. So how bacteria cope up with this double-edged sword? That's what we figured it out in our lab, that bacteria maintains two different populations within a quorum sensing population. So there are some number of bacteria which do not talk, and there is majority of the bacteria which talk. And we discovered reversible phenotypic heterogeneity as a bet hedging survival strategy. So I will give you a very simple example, then it will be very clear in layman's language. Suppose in your family there are 4 members, and one member is a bread earner, he is working. Second member helps in the cooking. The third member helps in household activities. But the fourth member does nothing. It takes benefit of all those 3, and he is surviving in the house. But now, suppose situation changes. For example, now there is a corona-like situation. Everybody is working from home. And now the bread earner also has to work from home. And while working, he has to use the computer. Now his computer fails. Now the fourth member, who doesn't do anything but very good in computer, chips in and helps the bread earner to fix his computer and to start the work. So this is what is called as bet hedging. So the population keeps variants or phenotypic variants so that they will take and do functions when the need arises. And otherwise they will remain silent in the population and take the benefit of the performers. And this is how the performer and non-performer looks like. You can separate them out in a bacterial quorum sensing population. So these are the non-performer and figured out, even if you take separated cells from either performer or non-performer, when you culture them back again, they again give rise to 80-20 ratio of performer and non-performer. So this is what is called as reversibility. This is very important because I told you in the beginning, in a couple of slides back, the reversibility is very important because if you see, imagine a scenario in which you allow cheaters to perform social tasks, other social tasks which are not performed by the performers, then soon the cheaters will outnumber the population and the population will be full of cheaters and they will collapse. So our nature keeps a hold on this process by having a switch of cheaters and non-cheaters in a regulated fashion so that 3 functions can happen simultaneously. One is quorum sensing by performing social tasks in unison and also keeping certain population of cheaters to perform specialized job when the need arises, and to keep a hold or control over the cheaters so that they don't overcompete and wipe out the performers. And this comes a very nice model which we gave, and this is the reversible non-genetic heterogeneity model in quorum sensing system. So this is we have experimentally shown also that in a biofilm condition, suppose at low cell density, the quorum sensing is less, it triggers, as the population builds up, it tells the bacteria to form a biofilm. So there are red and blue performers and non-performers. So red are the performers, blue are the non-performers. So the red one contributes towards the biofilm formation and the blue one takes all the benefit. But when the nutrients become limiting inside the host or in the environment, then the performers are tuned to stay in the biofilm because they are quorum induced, but the non-performers can move out of the biofilm in search of new niche. So they perform both tasks of migration as well as remaining in the biofilm to perform social tasks. Now I will come to you about how you can apply these strategies to counter diseases, both in human as well as in plant. So I will give you example of some of the plant health measurements by interfering with the quorum sensing system. So as I told you, that language system is very important, quorum sensing system, and it synchronizes the pathogen or the bacteria to perform social tasks. So both in plant and animal pathogens. So what are the strategy you can confuse the pathogen? Either you degrade the quorum sensing signaling molecules so that they are not able to talk, or overproduce the signaling molecule. So this is a very similar strategy when you are giving a lecture and somebody is playing music in a loudspeaker. So if you produce the signaling molecule in very high amount, then you will be breaking the communication system. So this has implication both in plant and animal health, as I told you, and the strategy to counter quorum sensing system. So there are two strategies basically. So there is a genetic engineering as well as a non-genetic engineering approach. In the genetic engineering approach, what we have done, we have developed transgenic plants which can produce the bacterial signaling molecule in very high amount, so that the situation will be like this. So when the bacteria will enter in those plants, it will feel that we are too high in number, we should not cause disease. But in India, genetically modified organism is not that popular. So we developed another non-genetic approach in which you can synthetically or biologically produce the signaling molecule in very high amount in other bacteria or in the same bacteria, genetically modified, and then spray it on the plants. Basically, you are spiking the plants with high amount of quorum sensing signaling molecule, so that when the next time when the bacteria infects, it will see the plant spiked up with the quorum sensing signal and it will not cause disease. So this is an experiment in which we have infected the plant with the bacteria which has not been spiked. You can see this is causing disease. But when you infect the plant with a bacteria, the plant which has been pre-spiked with the signaling molecule, it has stopped to cause disease. So this is coming up in as a large field, which is called as bacterial social medicine, which is called a social drug or antisocial bacterial agents. So in Pseudomonas aeruginosa, which causes cystic fibrosis, opportunistic pathogen, people in US, they are trying social drugs or chemicals which can either block the quorum sensing system or can block the iron uptake or social iron uptake. In our lab also, another part of our work is on iron uptake metabolism. In that we are trying this method. And social microbiology and developing antisocial drug is a very big scenario in Europe, particularly in UK. Many labs are trying targets or drugs which can block the cell-cell communication or social structure of the bacteria, because it is always beneficial. Because if you see the antibiotic scenario, so there is only one target and getting a resistance against one target is very easy. But getting a resistance against a social structure is very difficult because there are multiple circuits you have to mutate. So therefore, it's an excellent way of targeting pathogenic bacteria, which people are now working in a large scale. So at the last, I will show you how the bacteria talks. So this is a live cell imaging in which we took a single cell, and you see they are talking, and now you will see how they will be heterogeneous. So some will talk and some will not talk. So these are talking and these are emerging which are non-talking. And in that way, they give rise to a population in which there are performers and non-performers which can talk with each other and keeps a balance between performing social tasks as well as bet hedging to perform specialized tasks when the need arises, and also a regulated switch in which performers and non-performers are kept in a particular ratio. And these are the lab members in all the contributions are from our hardworking lab members, students. So all in red, they have left the lab. The graduate student, Ateeq and Shivashankar were the first student, Akanksha, and there were postdocs, Ananda and Narsima, Durga. They all contributed and talented project PDF, project JRFs, and they are the current lab members. We are still trying to understand the secret language of microbes, and I think it's a tip of the iceberg, and we are getting very interesting findings as we do more and more research. Thank you.
Thank you, Dr. Subhadeep. I think you have understood quite a bit about the communication, and wish you all the best for understanding much more about these communicating bacteria. We actually missed out your last two slides, but then okay. See, I think this all the viewers would agree with me that this was also an equally fascinating presentation with very beautiful, colorful graphics that very again lucidly explained the communication between bacteria. Talking bacteria, and we hope that it is good bacteria that does more of the talking. Now I will quickly peep into this our Facebook chat window and look for questions. There are many comments there. Yeah, perfect explanation, Dr. Bushra. That's a comment. Okay, here is a question from Shahid Aziz. He asks, how rest protein is regulated constitutively? This is to Dr. Bushra. I repeat, how rest protein is regulated constitutively? This is from Shahid Aziz.
You're on mute, Bushra. You'll have to unmute yourself.
Okay, good. So in prostate cancer cells, rest is not regulated consecutively. Like that, I won't say, because usually it's expressed in neuronal cells, and the main function is to kind of regulate the genes which are involved in neuronal differentiation. Only in prostate cancer, when the cells move towards NE-PC, at that time the rest level is gone down. And in prostate cancer also, like you see rest coming up, and then towards NE-PC it's gone down. So what strategy we use, what we have shown, that if you use a casein kinase inhibitor, so casein kinase is involved in degradation of rest. So it's phosphorylate the C-terminus dagron motifs of the rest, and as a result of that, it will undergo proteasomal degradation. So when we use casein kinase inhibitor, it will inhibit that degradation, and the level is maintained. And as a result of that, you see it's acts as a repressor along with AR, and SPINK is down.
Okay, the next question is, can selective androgen modulators be used for therapy for prostate cancer?
So, as I mentioned in my talk, there are several next generation inhibitors like epithelial-to-mesenchymal, and before that enzalutamide, and initially biclutamide were used. But all these anti-androgen, they have some resistance. Over a period of time, the patient developed resistance towards this inhibitor, and then the only choice is either block the synthesis or combine it with other therapies. So for blocking the synthesis, sometimes people use dutasteride, or even CYP inhibitor is used. Aberration is used, but again, majority of the patients, they develop resistance. So then they are put on chemo plus radiation, and obviously for various reasons they won't respond to these very what I will say extensive therapies, and then finally they succumb to extensive metastasis, and quality of life is also gone down, and then death.
Thank you, Dr. Bushra. I think the next question is for Dr. Subhadeep from Argha Das. The question is, how did you generate GFP-based biosensor?
So what you do is that you figure out quorum sensing regulated genes or the regulatory circuit, and you fuse your GAP, which has a very short half-life. That means the proteins, because you also have to take care that you use a GAP variant which is not stable, because otherwise you will listening to language which you spoke 1 hour before. So therefore you have to use a very short half-life GAP and insert it in the chromosome of the bacteria, and then your bacteria is tagged with the signal.
This is again another question to you from Dr. Geeta Vani. She asks, is quorum sensing known in viruses?
So far, quorum sensing is not has been demonstrated in viruses, but I will give you a very interesting example. Couple of months back, one paper came out in which they showed Vibrio cholera, which has been infected by a virus. That virus actually modulates the quorum sensing system of this bacteria. So the virus secretes proteins which can now modulate the quorum sensing system of the bacteria so that it will be beneficial for the virus to multiply inside the bacteria.
A question for you from Vinod Kochar, who asks, are the quorum sensing molecules characterized? And the second question, are they...
Many of them are characterized. Yeah, sure. Many of them are characterized. But as I told you, they are the tip of the iceberg. Because if you see in microbiology, 98 to 99% of the microbes we can culture in the lab, cannot culture in the lab. What we are dealing with just 1% or 2%. And when people do metagenomics of all the bacteria, culturable and non-culturable, people see diverse kind of quorum sensing signaling molecule biosynthetic genes are present. So in the 1% or 2% itself, there are variety of quorum sensing signaling molecules. Many of them have been chemically characterized, but in 90 to 95% of the bacteria which we can't culture, it's still a black box.
Okay, so those were the few questions thus far in the comment in the comment or chat window. Just to take a couple of popular questions, Dr. Bushra. So there are many types of cancers in India, and you chose to study prostate cancer. So is there a particular reason? Is it more prevalent?
No, my lab studies other cancers as well. So we are also working on breast, colorectal cancer, and glioblastoma. And we started with prostate. And the main reason was I had extensive experience in prostate cancer as well as in breast. But then when we moved back to when I came here and I found not much has been done in Indian context for the prostate cancer patients. So as I told you, the first characterization was done by my lab. And now we are moving forward. We are also doing more genomics. So next phase is to try to understand what are the mutations and what are the genetic aberration in context of by doing whole exome sequencing and other form of sequencing. And now with the recent this ND genome, few days back it was in news. So that will be a great asset for us to figure out what are the mutations which are more specific to Indian patient and how to design treatment for them. So as I told you, in West, most of the time these patients are given anti-androgen inhibitors, but in India still many hospitals they do surgical castration. So it will also be helpful to understand how the disease is progressing in context of drug resistance and what are the mutations which are the patient who are undergoing surgical castration versus chemical castration they're acquiring. So it's not that I'm only working on prostate, we're also working on colorectal and breast cancer and glioblastoma also recently.
Okay. Dr. Mande is on screen, sir. Do you have any question to ask, or even to Dr. Subhadeep? You can even ask questions to each other.
Now, I have a question about Bushra, that both in breast cancer as well as prostate cancer, BRCA1 and BRCA2 have some involvement. And is it clear that whether what kind of effect do they have on either breast cancer or on prostate cancer, these mutations, these genes? And are the
So yeah,
mutations common?
yes, so it's not, if we talk about in context of germline mutation, as I mentioned in my talk, like only 5 to 10% of the cancers, they're hereditary in nature, means they transmit from one generation to another generation. So BRCA1 incidence is very small, like it's less than 5% or sometimes 10% depending on the population that we are studying. So these mutations, they're not so bad, like they're mainly germline, and that put patient on predisposition to develop cancer. So in women, in mainly breast and ovarian cancer. In men, it's like prostate and other kind of cancers. But the good thing is, because in the BRCA1 is involved in DNA repair mechanism, so there are drugs where you could use like another DNA repair inhibitor, such as olaparib, like PARP inhibitor. So because of the synthetic lethality, they may respond nicely as compared to other patients. The other thing is like how prevalent they are somatically. If I say, but means like these mutation in cancer only, not in the germline DNA. There are like incidents where you see like a mutation of BRCA1 in breast as well as prostate cancer, but that is somatic. That may be a little bit higher.
Then what I wanted to ask you is that in all cancers, early detection is the key for better survival. And prostate cancer, unfortunately, the universal marker that people use, the prostate-specific antigen, is not that great. So what is the progress currently on having early detection markers for prostate cancers?
So that's a very good question, and as I also mentioned, so not gold standard biomarker, because sometimes what happens with age, there is enlargement of gland, and because of that enlargement, you see a lot of PSA coming up. And but you will say based on PSA level, it's reaching 10 or maybe higher than 10. People will say there's a cancer, but then when you take biopsy, it's not cancer. It's just because the volume is more and more cells are making PSA. So what I will say, like in the West, they are using a panel. So they're not relying totally on PSA. They're also including other genes that I mentioned briefly, like they're including TMPRSS2R, because there's only present in prostate cancer patient. But again, 50% will have, 50% won't have. So they're also including like PCA3. It's a long non-coding RNA. That is also a good biomarker. So it's a panel. And that's what I'm also discussing with the urologist and other medical oncologists in KGMU and AIMS to include a panel of genes. So my lab, as in the second phase, I was mentioning to Mahesh that we are coming up with the sequencing approach and come up with a panel which is more specific to Indian cohort, a gene panel where you include PSA, but there will be other genes also which are more prevalent at the early stage, so that we could detect and say, okay, this patient is on risk or not.
Yeah, thank you.
As of now, there is nothing. As of now, there is no system.
If you need any help in taking this forward, panels or sequencing or something, don't hesitate to contact us. We can actually help you in getting it forward and bring it actually in the clinical practice, at least the early detection. That's real key.
Thank you, sir. And I really appreciate the efforts put by IGIB and CSIR to make this indigenome, because before that I was like so worried how I'm going to rule out the variants, SNPs from the germline mutation, from the somatic mutation. Now because of that data, I think it will be very easy to rule out what is there in the Caucasian population, not to bother about that. Just see what variants we are getting which are specific to Indian population, and then see what I am getting, whether they're germline variant or like somatic or germline mutations. So thanks for that also.
So many of our younger audience would like to know from both of you, Subhadeep as well as Bushra, what motivated you to take up science, and what motivates you even now when you come to the lab every day? I see that you're in the lab right now. So if you can, both of you can comment on that one by one.
So I can say like I was a very curious kid. Like I was like, I love to catch butterflies and grasshoppers and grow some seedlings. And even like my mom, she was like a teacher in a government school, so her friend was a biologist, like biology lecturer. So Kumud auntie, I used to go in her lab and look at those specimen jar, in biology lab you see all those specimens sitting. So I think that kind of trigger like more interest in biology and the complexity of life, and somehow that got nurtured and I developed interest in that. The other thing is like what I'm doing, I love doing that. So that motivates me to do, and I don't feel it's kind of work. I mean, I love what I'm doing. So that itself is a motivation. And finally, it's like helping the society, especially the cancer patient. And I mentioned like every one of us, some acquaintance or maybe friend or relative suffer from this deadly disease. So doing little bit from my side make me like happy that I'm doing something like, you know, which is going to benefit the society at large.
Subhadeep, you would like to take?
Yeah, sure. So for me, it was the coming in science and working in science was by accident, because my father was in army, so he wanted to be me a soldier. So but my phenotype didn't cooperate. So therefore he gave up after that. So therefore, as I went to my bachelor graduation in Jamia, so I was quite interested in particularly the biochemistry and genetics, particularly microbial genetics kind of interested me a lot. And then since as a graduate student, we didn't got any much opportunity to work practically in the lab, and I got this opportunity in my PhD and also in MSc quite a lot. And that made me very interested in particularly the microbial biology and plant sciences, because animals, although I am a strictly non-vegetarian, but I can't handle non-veg preparation. So therefore, plants were quite fascinating. They don't cry and shout when you dissect them. So therefore, I landed up in plant sciences, plant microbe interaction, and in CCMB I got pumped up in Ramesh's lab. So working gave us a lot of freedom, and then this quorum sensing came up, and it's quite fascinating to work on system.
So curiosity and accidents.
We would have
curiosity, accident, and phenotype.
Yeah. I was telling Subhadeep we would have swapped the jobs, because after like my BSc and MSc, I kind of gave examination to join Air Force Short Service Commission in Varanasi. So I failed physical tests. Okay, I'll move on to PhD.
No, my father didn't allow for physical test. He saw my physics, physics, and told you better not apply.
Okay.
Because first of all, I was flat-footed. Okay. So that was one limitation.
Okay. On that note, I don't know if I can say this. You know, they say necessity is the mother of invention. If I can extend that a bit, curiosity is probably the grandmother for the quest of knowledge, and accidents. I don't know much about phenotype, but accidents do help. Okay, this I'm sure all of you who have joined us would agree that was exciting, informative, and enriching session. Thank you so very much, Dr. Bushra and Dr. Subhadeep Chatterjee, for joining us this morning. I thank the DGCSIR, Dr. Shekhar C. Mande, also for joining us, being with us, and driving this whole idea of webinars forward. I also wish to thank Dr. Geetavani, Head of the Science Communication and Dissemination Directorate at CSIR, and our team members for helping with streaming and other support. Finally, and importantly, to all those of you who are connected with us, thank you for joining us and interacting with us. The recording of this session will be available on our social media channels. And do join us for the next session of Exploring Science with Shanti Swarup Bhatnagar Winners 2020. Please connect with us through our social media pages for the announcements, updates, and see you all very, very soon. Goodbye for now. Wear a mask. Stay safe. Namaskar.
Thank you.
Thanks.